Bismillahirrahmanirrahim. Welcome back, friends. Welcome to another video lecture from Kami Biology. In the previous video lecture, we talked about in a detailed reverse transcriptase inhibitor drugs. Fine. Now this is the last video lecture regarding with antiviral drugs. And in this video lecture, we are going to talk about the last drugs, and these are the neuraminidase inhibitor. Now the drugs involved in neuraminidase inhibitor are Oseltamivir, zanamivir, lenamivir. Right. So now let me write. Neuraminidase inhibitor. The drugs which are used in the neuraminidase inhibitor, these are zanamivir, Oseltamivir, and lenamivir. Fine. So these are the drugs. Now let's talk the antiviral spectrum of these drugs. These drugs actually show good result, or these drugs is activated against influenza virus A and B. Influenza virus A and B. Now what are the mechanism of action of these drugs? So let me write. Mode of action. These drugs actually inhibit the neuraminidase enzyme. Now what are the neuraminidase function and what actually they do? Let me explain all this with the help of diagram. In this case, let's suppose we take the influenza virus, because these drugs are activated against the influenza virus. Let's suppose this is the envelope area of the influenza virus. Fine, because influenza virus is the envelope virus. This is the capsid area of the influenza. This green area represents the capsid. Fine. Inside this capsid, they carry the negative single stranded RNA. Fine. Now there are some protein channels are arises. We usually call that M2 proteins. And there are, now this black one represents what? This black one represents the neuraminidase enzyme. And within with the neuraminidase enzyme, there are some other protein like structure, and usually we call that hemagglutinin. And after some hemagglutinin against this neuraminidase enzyme are present. Fine. So these neuraminidase are present on the envelope area of this influenza virus. Now this single stranded are within this single stranded RNA, there are polymerase enzyme present. So this is the polymerase enzyme. Usually we call that RNA polymerase enzyme. Actually there are RNA polymerase enzyme present. Whenever they are entering inside the body of the human, and then they are finding a host cell. So if I draw the host cell here, so what happened actually? This is the host cell. Means this is the host cell surface, or we can say this is the membrane, cell membrane. Now within this cell membrane, there are some silica groups attaches. Now this structure represents the silica group. These silica group are actually responsible to attach these hemagglutinin. Let me write. This green one represents the hemagglutinin. Hemagglutinin. Maybe the spelling is right or wrong, but then you will check the after the lecture you will check the spelling whether it is right or wrong. Now what happened? These hemagglutinin are actually, these are actually spikes protein, we can also say. they are attached with this silica group, which are these silica group actually represent the receptors on the host cell. Now, due to this silica group, these are actually although they are not receptor, but these are used as a receptor for this influenza virus. What actually this influenza virus do? This influenza virus will attach the hemagglutinin to this silica group. So once they will attach to that area, then what happen? This neuraminidase enzyme, let me write, neuraminidase. So they have two kind of activity. So now let me write. One, they have exoneuraminidase, and other they have endoneuraminidase. Fine. So they have two activity, exo or an endo. What happen? This neuraminidase enzyme will first show endoneuraminidase activity. Now what happen in endoneuraminidase activity? They will cleave the silica group which are present outside this host cell membrane. Now with this host cell membrane, this silica group will be cleaved by this neuraminidase enzyme. So this is the endoneuraminidase activity. So once it is cleaved, then the cell will be then the virus, this influenza virus, will be internalized, fine, through a membrane fusion. So once they internalized, then they will release their genetic material. I already discussed the replication scheme of the negative viruses, that how negative viruses means negative single-stranded viruses, that how they are replicated inside the host cell. And so I already explained it. So go back to my channel and watch the video. Now they will internalize and they will start their replication process. So once their replication process is completed, let's suppose the replication process is completed, and what we get this influenza virus again. So this is the capsid area. This is the this is the envelope area, and this is the capsid area. Fine. So within the capsid they carry the genome, negative single-stranded genome, and outside there are these neuraminidase enzymes arises. With this neuraminidase enzyme, there are the hemagglutinin. So what happened after that? So once this neuraminidase enzyme are mature, and once this neuraminidase enzyme are formed inside the host cell, so let me draw the host cell in. So let's suppose this is the host cell. Now neuraminidase are formed inside the host cell. So this is the host, or we can say mammalian cell. So let me write here mammalian. Mammal or host cell. So once they are formed, so they will released. Now how they are released? These actually here inside the host cell there are also the silica group are present. So this silica group are not only present outside, this silica group also present inside. Now what actually happened? This hemagglutinin again attached to that area, this silica group. So once they are again attached, so this neuraminidase enzyme, then this neuraminidase enzyme will show exoneuraminidase activity. And what happened in exoneuraminidase activity? Due to the function of this exoneuraminidase activity, they will cleave the silica group which are present inside the host cell in this membrane area. So once they are cleaved, so again the cell will the virus will release through a membrane budding. So again these viruses are released out from the host cell. So they will release out because this neuraminidase enzyme will cleave this silica group. Now what actually the drugs do here? Means what are the function of these drugs, and what are the mode of action of these drugs? These drugs, let me draw here, these drugs will bind to that area. This red one represents the binding ability of the drugs. So this is the drug. So the drugs will bind here. So once the drugs will bind here, so here what happen? This silica group will be not cleaved. So once the silica group is not cleaved by the neuraminidase enzyme, means when the silica group is not cleaved by the activity of neuraminidase, so this means this hemagglutinin will just attach, but due to loss of the function of this neuraminidase by the drugs, means here the drugs blocked this neuraminidase enzyme, then this virus will be not internalized. This virus will remain outside. Fine. So this virus will be not internalized. And let's suppose the virus will internalize and the virus are formed. Now the second activity or the second mode of action of this drug, what happen? These drugs will enter their self inside the host cell, and inside the host cell these drugs will again bind to that area. So they will now, in this time, they will block the exoneuraminidase activity. So here exoneuraminidase activity are also stopped or also halted by these drugs, mean by these oseltamivir and zanamivir. Fine. So they will again, although they are attached to, means this hemagglutinin attached to this silica group, but these silica group are not cleaved by this neuraminidase enzyme, because here the drug blocks it. The drugs block the activity of this neuraminidase enzyme. Fine. So this is the mode of action of these drugs. Now, what are the resistance of these drugs? What are the resistance which are activated against these drugs? The resistance which are activated against these drugs in a 14-year-old girl. In a 14-year-old girl, means in a 14-year-old girl in a country of which name means it is the name of country. So she lived in this country, and once the scientist isolate the viruses from his body, then what happened? This is also influenza virus. Means whenever they isolate the viruses, so these viruses are also the influenza viruses. But here means this neuraminidase will show different means at this neuraminidase enzyme there are some positions, means at 274 position there are the enzyme we call that histidine. Fine. So when at 274 position, the not histidine is actually amino acid. So this histidine are present at 274 position in the neuraminidase enzyme. So once the histidine are present, so this drug will be show their activity. What happened? This neuraminidase will change this amino acid to tyrosine. Now here the histidine amino acid is changed into tyrosine. So once the histidine is changed into tyrosine, then the drugs will not bind here, because the drugs are bound here because here the histidine are present. That's why the drugs show activity. But if the histidine are substituted or are replaced by the tyrosine, so then this influenza virus will show resistance against these oseltamivir, zanamivir and lenamivir. Then these drugs will be not affected here. Right? So this is the means at that area there are some position. So at 274 position there will be the tyrosine. So that's how they can show the resistance also, and the resistance will be shown in the 14-year-old girl. She actually lived in the Vietnam. Right? Vietnam is actually the name of country. So, and when the scientists isolate the viruses, so these viruses are actually what? These viruses are actually influenza viruses, but this influenza viruses we call that H2N1 influenza. This influenza virus, this is the strain of influenza virus. Now this influenza virus strain have tyrosine instead of histidine. So that's why this influenza virus show resistance. So this is the resistance. Now let me talk the adverse effect of these drugs. So the adverse effect of these drugs are the GIT disturbance, asthma and nausea. So that's all about the neuraminidase inhibitor. Fine. The resistance mechanism of action of these drugs and the adverse effect. Now this is the last video, and I hope all these videos which are regarded with the antiviral drugs will give you a brief idea that how actually these drugs will show their activity against the viruses, what are the mechanism of action, what are the resistance, and so you will understand in a very detail. If still you have any kind of question about these antiviral drugs which are which contain the 7 videos or 8 videos, so write it into a comments. I will try my best to give you answer as soon as possible. Fine. So I complete my work and I do everything for you, and I explain the lecture in a very detail. So now it's your turn and it's your number. So please like the video, share this all videos lecture which are regarded with the antiviral drugs with your friends. Fine. So, and also subscribe the channel for upcoming videos in a detail. So thanks a lot.